Good morning. Welcome to the workshop on integrative models of aging core. And today we will tell you about the services that are available in the core. And there are two leaders in this core, Max Shokhirev and myself. And we will tell you about the methods that we offer and for the analyzing of um, data sets. Okay, so Max will start and I will continue.
Right. So yeah, let me share my screen too.
Okay, so you want like this? Okay.
Great. Can everybody see?
Yes, Mike, it was good.
Okay,
Are you on your first slide? Yeah.
Yeah. Thank you for for giving me the chance to talk about our core. Basically today, I'll kind of go through and give you a 10,000 foot overview. So first, I'll kind of go into the challenge of studying aging, the different technologies that are available. I'll introduce the team, talk about the services that we offer, and also give you kind of an example project to give you kind of a feel for how the interactions work. And then I'll pass it over to Tatiana to talk about her hierarchical data visualization methods. And then we should have, at the end, some information about how to get in contact with us and also a Q&A session. So, as we all know, aging is an exceptionally complex biological problem. It basically affects pretty much all cellular processes. So this is just kind of an old review now of all the different kind of hallmarks of aging. I think there's probably a lot more now. And also, aging is heterogeneous, right? So not all cells will age at the same rate. So if you look at isotope microscopy, you can see that some cells actually are very long-lived. In this case, this is in the pancreas. And then others are turning over pretty frequently. And we also know that aging is organ specific. So each organ has its own aging phenotype. There's some global patterns, but also there's organ and tissue specific patterns. And we know that there's interactions between organs, right? So whether that's through the blood or through the immune system. So this is actually a very complicated problem to study. And then, of course, that leads to variability between individuals and populations. Right? So how do we approach this? Well, as you've already heard, there's a lot of really exciting technologies that are available and are kind of maturing. So there's high throughput sequencing, right? So this is kind of the standard kind of bread and butter that people use. So you can look at the transcriptome, you can look at where proteins bind, you can look at chromatin accessibility, single cell sequencing, of course, single cell RNA sequencing, single cell ATAC, also spatial transcriptomics now. Then there's also mass spectrometry, so proteomics, metabolomics, and of course microscopy, as I've already mentioned. So you can look at age-associated features. We get a lot of really high-resolution images. And what's nice about this is that you get a lot of data, right? You have a lot of data, and it's it's very high throughput. So you also need a way to store everything and make sense of it. So for the core, we have about a petabyte of storage and 9 compute servers, as well as cloud. So this is all available to us at the Salk data center. But of course, once you have all this data, this is now a different type of problem, right? So there's so much data that, what do you do with it? How do you make sense of all of it? So this is kind of the main motivation for our core. The data is being generated. Now, how do you put it all together and gain some sort of insight into the underlying biology. So we do this by establishing collaborations with biologists, and we offer a whole range of different services. Everything from, at the beginning, come to us, let's talk about, let's plan the experiment, figure out what your biological question is, help you kind of determine what kind of controls do you need, what kind of replicates, what are the limitations, timelines, things like that. We also have, as I mentioned, data storage and curation. So everything is stored in redundant kind of backed up servers, and can also help with online kind of submission to repositories. We perform quality control to make sure everything is technically sound. And then we have many different types of standard and customized analyses. So pretty much anything you can think of, or we can implement it. And then the big thing is, how do you put everything together, right? So how do you integrate everything? So we can do this with different mathematical models, different tools that are available, machine learning. Tanya will talk about her methods. And then, the other thing we can also help with is kind of building these online tools. So if somebody wants to interact with your data sets, we can help with that. And finally, training, right? So training, these types of workshops, one-on-one training, we provide materials. Really, the idea is to be very collaborative, right? So if you reach out to us, we'll help you. And we have a dedicated team. So Tatiana is, she's excellent and can, help with any kind of, math question you might have, statistics question. I help lead a crack team of PhD level bioinformaticians. And we've co-authored over 60 publications in the last 4 years on like a wide array of different topics, including 14 aging papers. So we kind of seen a little bit of everything at least. So I think the last thing I want to kind of go through is just an example of a project. So this was a kind of a very interesting project. Antonio came to us. He had this interesting data set. He had these premature, mice that age prematurely, and he wanted to figure out what's happening with them. And one of the questions that he had was, if you add these specific drugs, they start behaving differently. They start looking like they might, be reversing their cognitive decline. But then the question was, how do you, what does that mean in terms of the mechanisms? Right. So, how does old age affect gene expression and metabolism? And then what do the drugs do? And he had 2 different types of assays. He had RNA-seq and he had metabolomics. And then he had middle-aged and old-age mice, and then also old-age and drug-treated. And Ling kind of teamed up with him, and she helped him with the RNA-seq analysis. And one thing that stood out is that basically genes in the mitochondria are dramatically downregulated, and they're affected by these drugs. So he went out and performed this metabolomic screen, and then we kind of manually looked through this, and he manually looked through all of the different metabolites, and Ling helped him with visualizing everything in terms of a network. And I think at the end we were able to kind of pin down this acetyl-CoA pathway that was affected. So this was a really great collaboration, and it led to a nice paper. But Ling actually was kind of inspired to then kind of make her own tool, right? So the tools that we have are kind of, they weren't that great. So she developed a novel multi-omics tool working with Antonio, and he actually ended up measuring proteomics as well. So we have transcriptomics, proteomics, metabolomics. And she developed a tool that kind of puts all of that together into a network-based model with actual reactions being simulated. And all of this is now an online interactive tool that people can use. And then she went back into that data set. So before we were able to see some changes in the metabolites, but it was kind of, not that many, and also kind of, difficult to figure out how they all connect to each other. But then if you apply this kind of integrative method, you end up seeing a lot of differential reactions. And there's overlap now between the changes in the old versus the young, and then the drug treatments. And so this kind of enabled her to go back and reanalyze that data set and really form additional connections using this tool. And they're also, they're relevant to the TCA cycle and the, acetyl-CoA pathway. So I think with that, I'd like to kind of stop and invite you to come surf the data tsunami with us. And if you have any questions, please write them in the comments or the chat, and we'll be able to come back to them and answer them at the end of the session. And I think with that, I'll pass it over to Tanya.
Okay, I will continue. So with more description of on mathematical modeling, and maybe not with existing tools, but with more custom-based tools for data analysis. And the kind of current state-of-the-art methods that we are developing, sometimes not, some of them are in the stage of development, is identifying causal interactions in networks and characterizing hierarchical data. And as Max pointed out, identifying driver nodes with new and additional methods that I will describe. And also visualization methods for hierarchical data. And what we're also actively working with various groups is custom methods for quote unquote batch correction. And what we've seen sometimes is that taking into account these hierarchical arrangements of data leads to very good results in that respect. So in general, our approach is kind of what I think is a novel approach, but it's gathering also a lot of interest from across the country, is that when you have a hierarchical data, like on the left here, the way we compute distances, or maybe it's not a perfect hierarchy, maybe it's an approximate hierarchy, maybe there are loops in the network. So how would you evaluate distances if you want to put data on a map, on a continuous map, not necessarily on a discrete map? So we need distances. And if we want to evaluate distances between points A and B, then basically we have to go towards the root of the tree and then back out. And what this leads to is that the trajectory that connects the nodes becomes curved. And one can approximate this trajectory using the so-called hyperbolic geometry. So it's the same geometry that you may have heard describes our universe on the grand scale, but we also see that the same geometry applies within cells and within the organism to describe data within these little hierarchical domains. So this is an example representation on the right using a continuous representation of data. And you can see there are more nodes that kind of generally, there are fewer driver nodes and more daughter nodes at the edge of the space. And here would be an example of trajectories that connects them, the shortest path between the nodes. And this is maybe very abstract, but I will show you how we use it in one could say, metabolomic data set from plants and from animals in the olfaction, various secondary metabolites that were produced by fruits and also mice. So what we find is that typically many kinds of data sets, instead of a kind of a two-dimensional network, are better described with a three-dimensional network in the hyperbolic space. And other groups are also developing, one can use this visualization for the tree of life and actually very efficiently put all species into a three-dimensional hyperbolic tree. And to visualize this is, if you take one way to visualize it is imagine an envelope for this tree. So the space is not spherical, like we live on Earth, so we think that the distance across different cities on Earth will be along the surface. But here, imagine this is like a sphere. This will be an envelope to the data. But to measure points, in this case, there are different orders that were produced by plants. We will go inside the sphere, and that will be the path that connects different molecules. So this is one type of representation. And in this case, because they were all volatile molecules, they all sit on the surface because they're all the consequences of what happened inside the plant. So we can infer which pathways were activated based on the secondary signatures. And here is an example of a different visualization now from phenotypic measurements in humans of the same kind of order molecules. And in this case, we see that you have points both within and outside of the space, and one can identify more driver nodes would be closer to the center of the space, and more daughter nodes on the surface. And so one can, using these coordinates, you can assign how close each node is, how high in the hierarchy of influence. So we also, using these methods, if we find that taking into account this hyperbolic geometry leads to quantitatively better visualization of the data compared to standard methods such as t-SNE. Many of you have heard of it, or many maybe have used UMAP. So these are quantifications of how the data is, distances are preserved after the low dimensional embedding. And in this case, this is just the general gene expression data across different types of human cells. And one can see that, for example, compare UMAP representation versus where the clusters are not, sometimes it's over clusters the data, and also the position of the clusters are not, are somewhat arbitrary. So if you put them on a kind of global map, then the position of the clusters is more constrained. And what is also interesting, and if we focus on this cluster of magenta cells, so these are breast cancer cells, so one can notice that some of these cells are closer to the center of the space, and we think that these are more dedifferentiated cells, so maybe cancer stem cells here. So one can use this method as a diagnostic for identifying kind of subsets and characterizing heterogeneity in the data set. So these are all methods that are available as part of the core, and we are working with, I'm happy to work with you on the development of methods that are suitable for a specific data set. So this is another example, in this case for blood cell development, where you can see continuous pseudo-time trajectory estimation with our newest, latest and greatest methods of this in this hyperbolic space based on t-SNE, but with hyperbolic metric compared to other methods, if you like to use. They're called Poincaré map from this publication here. And in this case, you have a local hyperbolic geometry, but global structure of the t-SNE method. And we see that the quantitatively, this is a better representation as quantified here and more continuous in terms of pseudo time if we impose large-scale hyperbolic metric and and locally kind of more standard Euclidean metric. So in summary, what are the methods that are currently available in addition to the ones that Max mentioned? We can use standard method, t-SNE, UMAP, PCA, but also with different types of metrics such as hyperbolic t-SNE, hyperbolic MDS, and other methods. And other methods for geometry detection and dimensionality reduction, and methods for identifying driver nodes. So with this, I will finish by putting our contact information here, my email address and that for Max, and ask you for your questions.